Our signal represses the activity, the tumbles. The methylation increases activity, but X inhibits. This is our negative feedback loop. Why? Because X activates B that removes the methylation. So this is a negative feedback which has a slow and a fast arm. And it's like that slow, fast, negative feedback I mentioned in a previous lecture is a motif for controlling, for adaptation. Just like a heater, slow heater and a fast thermostat can reach the desired temperature.
So about some level of attracting to methylation will be at its maximum and won't be able to deal with this anymore, or is this?
So the question here is about if methylation reaches its maximum, if all the receptors are fully methylated, then we have a problem. For some attractants like serine, that's a real problem. So if you have very high levels, receptor fully methylated, and they can't adapt anymore. But for aspartate, that doesn't happen, so it doesn't max out. This is important. All right, so we have our more complicated protein circuit than we've ever seen. We have the forward arm with the phosphorylation, the backward arm with the adaptation, the negative feedback loop. And the final question I want to address here is about robustness. So I say if you try to model this, you typically can get exact adaptation, but you have to tune exactly how much R and B there is exactly so that the drop in activity is exactly compensated by the rise in methylation. And then if you change R, let's say, by 20%, only a 20% change in one of these proteins, you lose it. You lose exact adaptation. You don't go back to baseline. And this is typical for almost all circuits you can write down. But in bacterial chemotaxis, you have exact adaptation. And if you change R, B by a lot, you can change the steady state level, but you still get exact adaptation. Robust, exact adaptation. So this is called robust. And circuits where you need to tune things, protein levels, are called fine-tuned. They can work on paper, but they won't work in the cell, because in the cell protein levels vary. Okay. How does this work? And how does robustness work? Maybe I should say in my postdoc, I did these experiments because my goal was to test whether exact adaptation is robust. So what I did in my postdoc was genetically engineered E. coli, and I forced them to have more or less of each one of these proteins, X, Y, R, and B. And the way I did that was I deleted, let's say, the gene for R, so the cells can't make R anymore. Cells that can't make R anymore, by the way, are completely unmethylated, therefore have zero activity, and therefore they just run. This is like example bacteria, they never tumble. And then I added this R on a plasmid under a sugar-inducible promoter. So if I add the sugar, basically IPTG, I could make a desired amount of R. So I could change R. And when I changed R, I observed that the steady state level changed. So instead of 1 per second, let's say now they're at once every 2 seconds. And I add the attractant, they respond, but they go back to their base level. So what I discovered in my experiments was that steady state level is fine-tuned, depends on protein levels, but the property of exact adaptation to whatever the steady state level is robust to all the proteins. It's changing factor 2, 3, 4, any of these proteins, you still get exact adaptation. Did I explain myself? Yeah.
Just taking you back a little bit. So if R and D change independently, then we will lose exact adaptation? Is the fact that they change in some way related to each other that they keep seeing an exact adaptation?
No. The question is, did they change independently? In my experiment, I changed them dependently or independently, and you always get exact adaptation. It's not dependent on a proportional change or something like that.
So when do we expect to lose exact adaptation?
This is in models that try to model what I just told you mathematically. And it turns out it's hard to do this. I want to show you a model that does do it. So in the book, there's a full solution of the first model for bacterial chemotaxis by Lee Siegel, Albert Goldbetter, very important model. And that's basically the model for exact adaptation used for a few decades. But they didn't ask this question of fine-tuning. So they were fine with having the phenomenon for certain parameters. But if you add the knowledge that these parameters, basically protein levels, change from cell to cell, you realize that you need that's a constraint. That model and many other models don't work. You need to have zero in on a very special model. Not only gives you exact adaptation, it does so for any parameters, any protein levels.
But this model is the one that needs to be fine-tuned to produce exactly.
This is not a model yet. This is just biochemical knowledge.
Okay.
Now we're going to write a model, the equations. The model for me is equations. The different fields have a different meaning for the word model, like in fashion, for example. Or in biology, a lot of times this is like a model, right? But in biochemistry, but now we're talking mathematical model. Okay. Oh. You know what? I want to do something before I do this model. I want to recognize this is a complicated bit of biochemistry. So I want to, instead of getting a lot of questions, I want to propose that we'll use the heterogeneity of this class to ask you to talk to the person sitting next to you to see if you can understand the response and adaptation here in terms of this methylation. You find that if you try to explain it or ask questions, you have more chance to figure it out, right? So you don't have to, but I recommend turn to the person sitting next to you, and we'll spend a couple of minutes, see if you can explain this methylation, see if there's still something to understand here. So please go ahead and do that. Or in threes you can do, or look behind you. בסדר? רוצה לדבר איתו? Okay. As I look at you, so maybe I made a mistake in this, or just want to make sure from you, what are the questions that remain? Yeah.
You have here that X has two ways to regulate the methylation. X star, the bottom graph on the board.
This one?
Yeah.
Yeah.
One fast and one slow.
So sorry, these are both. This thing here, what I meant to draw here is just an explanation of this arrow. It's an so X activity reduces methylation by means of activating the protein that removes the methylation. That's the meaning of this. So this is a slow arrow. This is an explanation of this arrow. Yeah.
Again, so
X
is removed.
active X activates B. I'll tell you soon how. Therefore, reduces methylation.
Right.
So, so this is the actual reactions plus minus. Okay, so this is a negative arm, and here M, as I told you, increases activity. So it's a positive arm. So this is the way it works. And this is, by the way,
So the more methylation you have, the more active X for
Yeah.
a given amount of S.
yeah. And now I still need to explain you that biochemistry. How does methylation increase activity? So, so remember I told you that the receptor activity drops with S and has this halfway point K, where it's halfway down. So the more signal, the less activity. This is what the attractant does. What methylation does increases this K. And so methylation increases K. So K basically shifts here. And so what happens is that if I have some signal, let's say I have a signal, I have some attractant here, this is my activity. I now move to more signal, I drop in activity. And now slowly, methylation goes up. K moves over here. So this is the curve now. And activity goes back. And the way that K in methylation increases K is also known biochemically. K is basically given by the free energy of the active and inactive conformations. And what methylation does, each methylation adds a little bit of free energy. So in fact, K goes exponentially with methylation. And that's the way that the methyl groups changed. Basically, they changed the charges on the protein so that the free energy of the active and inactive state changes. And each methylation adds more.
They lower the free energy of the active state.
Yeah, they lower the free energy of the inactive state, actually. Of the active state, of the active state. Yeah.
It pays more
Yeah.
to be in the
Yeah. That's how it works. So we have like a full biochemical understanding. So this is, the more methylation, the more K shifts. So that's the way it works over there. Yeah. Okay, now I want to write a little, again, the model will be very simple. Everything we heard so far is much more complicated. The biology here is ingenious, and the way it was discovered by people like Mel Simon and Koshland and these great biochemists that work on the system. It was the first two-component system to be analyzed, and it was basically one of the first signaling systems that basically this knowledge later helped us understand human signaling and cancer, etc. Very important system. But the math, I think, turns out to be quite simple. So the people who figured out how to have a robust mechanism are Nama Barkai, our very own Nama Barkai here from Molecular Genetics Department. When she was a postdoc with Stan Leibler. And Nama came to Stan 1 year before I did. So she did this model. And the reason I did those experiments was because I was so excited about the model I wanted to experimentally test. Is exact adaptation really robust? That's why I changed from a theoretical physicist to an experimental biologist and learned how to make all those plasmids. all this microscopy to change key R, key B, key Y, key Z in the cell, measure the response to steps of attraction. See is what changes, right? As I told you, steady state activity changed when I changed protein levels, but still there was exact adaptation to that activity level, steady state activity level. So you can have cells that tell them serve 1 per second, twice per second, but they return to this twice per second. Did I explain myself? So how this? But before that, Nama worked out this model. So this was again a revolutionary step in system biology or biological physics because it was the first time also that it was understood how this question of robustness was basically raised in this context of signaling networks. And this is now a very big field that's there. So the way it works is it needs two things. One is that R works at a constant rate. Basically, it always has some substrate independent of how many X unmethylated. It always has some place to put its methyl groups. And the second thing is, this is very interesting, this B that removes the methyl groups only can do it when the receptor is in its active conformation. When the receptor is active, the methyl groups are pointing outside, it can grab them. B only works where it removes the methyl groups from active receptors. So receptors are active, inactive, and this B is a protein that can recognize the active conformation and take away the methyl group. Okay, these two assumptions lead to the following equation. So I'm going to write the rate of change of total methyl connected bound on the receptors. So this is the MDT. This is how many methyl groups. And it's added and removed, right? So it's added by key R at a constant rate. VR is the rate per R protein. So this is number of R proteins times the rate. Yeah? And it's removed by B, but B needs to collide with an active receptor. No matter how many methyl groups it has, whatever, it has to be active receptor. Take away the methyl.
How is it that the addition of methylation is independent of X because R is not specific?
Because R is at saturation. So most enzymes, if this is the substrate, what they're working on, and this is the rate, it saturates like that. So if you have so much substrate, they're basically working at their maximal velocity. Substrate here means unmethylated receptors. So there's 100 key R, and there's 10,000 receptors. And they always they're saturated, basically. So this is what's called zero-order kinetics.
But does this solve the question why when X is not active, it works at the same rate when it is active? Because also when it's active, it exposes the sites of the methylation.
As far as we know, this function of R doesn't depend on activity.
Which is kind of weird, right? Because it physically exposes
Yeah, but it actually needs to find a place to methylate. So there are 5 places where you can methylate, and it doesn't care, basically, whether they're inside, outside. But you can add here also 1 minus X star or something, like only on the inactive receptor. And then what I'm telling you still works. But, all right, so this was just an assumption, by the way, back then, but now it's biochemically later shown to be correct. Well, yeah.
How do you account for different degrees of methylation?
So B doesn't care about, just cares if it's active or inactive. I should tell you that completely unmethylated receptors are always locked in the inactive conformation, so that's not a problem, but because of this free energy. But usually receptors, when they're made with special modifications that look like they're made with 50% methylation, more or less. And so that's the steady state, is around 2-3 methylation groups. sites on the protein that are methylated.
You don't account for maximum amount of the methylation?
You don't? Maximum of methylation, if all the receptors are methylated. Then it's even better for B because then it has lots of substrates. So,
Or the other way?
yeah. You can put here any increasing function and it still won't tell you will work. So you can account for saturation. Anyway, what am I getting at and why am I teaching you this? Because this equation, I'm going to rewrite it here. I'm going to take VBB out of the parentheses. And then what I get is VRR divided by VBB minus X star. And I want to tell you that the steady state of this equation, so the steady state, so when dm dt equals 0, there is no more change in methylation, is when X star equals X star steady state, which, you know, if you solve this equation, VRR equals VBB X star divided by VBB, is basically the ratio of the action of VR and VB. So this is the steady state. And what we have here is methylation looks at the difference between the current activity and the steady state activity. Okay, so the only steady state possible is when X star activity goes back to this X star steady state, which is dependent on the proteins, but not on S. So no matter what happens, what R and B is, I put attractant. Methylation is going to start to increase. Why? Okay, I put attractant. I put on attractant, activity immediately lowers. And then this thing here is positive. X is below its steady state. And methylation starts accumulating. And when does it stop? It stops, it can only stop when X star reaches its steady state because of that equation. So before I had X star steady state, and after I had X star steady state. The value of the steady state can change if I change key R and key B proteins. X star steady state can change. But whatever those proteins are, key R and B, I'll always get a return to the steady state. So if I change, like I did in my experiments, that's what I saw. If I change R, I change steady state by return to that same steady state. So let's say this is, by the way, what happens when you lower R by 2.
Can I ask a question? How can this be defined? I mean, I understand there is robustness with respect to the shape of the function, but how can this be defined as robustness? Because I mean, it does matter very much what is the steady state, because it influences the frequency of tumbling.
Thank you. So you're right. In this circuit, and just if we're going to spell it out, we say in chemotaxis, the steady state tumbling frequency is fine-tuned. It depends on the protein levels in that particular cell. But the property of exact adaptation is robust. At the same time, there's two features. One is fine-tuned and one is robust.
So if you have some ratio, the ratio is what matters. If you have some ratio of R and B,
then you're fine.
Regardless of
Yeah,
robustness,
yeah.
then the cell might still be in a lot of troubles because it cannot reach some sweet frequency of tumbling.
Uh-huh.
Is it, or is it wrong?
Well, I'm going to talk about that in a second, okay? This is the last point I want to make. Before that, I just want to say that this way of writing an equation has a name in engineering. So engineers, 150 years, use the same principle, and it's called integral feedback. And the idea is that you have some integrator that, or something that integrates the error. The error is x star steady state minus x star. So the rate of change of m is this difference. This difference, we can call it the error. How far away from steady state? And m basically integrates over this error. And then m increases activity back. The only time, the reason this is such a powerful feedback is that as long as you have any error, as long as the x star is a little bit different from x steady state, this integrator will keep working. So it's a very powerful. And that's what engineers use in this room. This is with this thing here. So here, at the beginning, you asked me to put on the air conditioner. So I punched in here, 25 degrees. This is the steady state that I want. By the way, how many of you here know what integral feedback is? Heard about it. So not very few, because it's basically used in almost every device that's controlled. Every airplane that you want to stabilize there. And I just want to walk you through. You'll see it's the same equation. So we have a heater, okay, that has a power P, and a room temperature T. And you know, how does the temperature change? First of all, if we turn on the heater, it's going to make some heat and change the temperature. And then, if I open the window, I add additional sources of heat. Or there's a lot of people in this room, each one makes 100 watt extra source. So that'll increase the temperature. Right? And there's a coupling to the outside. This is how Newton's law, right? Temperature diffuses out. And I don't necessarily know these numbers. I don't know how many people are in the rooms, but I still want to get 25 degrees. How do engineers do that? They put a controller, that thing there, that says that the power, there's some rate constant, is T steady state, 25 degrees, minus the actual measured temperature. This integrates over the error. So let's just check it out. Suppose the temperature is too high, more than 25 degrees. What will happen to the power? It'll go down. Temperature will go down. So I want to suppose my temperature now is 30 degrees. Okay, it's too hot for me. Okay, so this is minus 5 degrees here. Power will go up. Temperature. Oh, what did I do? There's I looked at a heater, but this is actually an air conditioner, right? So I turned the sign. So if you turn the signs out, you get it's not nice, right? You get a disaster. So what should I do here? What should I do? Should I put a minus here? This is going to be, or we're going to talk about a cold country. Okay, so many of the viewers here. Now it's April in Israel. It's so nice outside. Right, May.
You could just put a minus before that.
Yeah. So let's talk about a heater. Let's talk about a heater. Okay, heater, not an air conditioner. Right? Heater. Yeah. Thanks. Okay. So let's say I put 25 degrees, and now the temperature is 15 degrees, right? Yeah, the sign is here. Okay. So temperature is too cold. Yeah? So this says increase the power. So things get high, hot. When will it stop? When temperature equals 25 degrees. Now this equation will always stop at 25 degrees. If my heater is getting old and it's coupling to the room, or I open up a window, or I put more people in the room, or I change the thermal coupling, this doesn't matter. Any parameters here don't matter at all. The only thing that matters is this T steady state that I put inside there. And this will only stop always at 25 degrees. power will compensate for changes in any of the C1, C2, C3. So that's the way integral feedback works. So I say if my temperature looks like this, and my power looks like this, and now I open window, and temperature goes up, or let's say I open a window, it's cold, so temperature goes down, power will go up, and temperature will go up, and return to this 25 degrees, and power will now stay up, just like methylation. This is like methylation, and this is like tumbling frequency that we can control. So this is exact adaptation, and the set point is inside the cell, is the ratio of these two proteins. So bacteria discovered using enzymes how to implement an integral feedback, which is such a powerful feedback. It guarantees that for any step disturbance, you return to steady state. And it guarantees it, and this. I want to mention Ian Doyle, who took the Barkay-Leibeler model and mapped it to integral feedback to look at the general principle there. And since then, I would say integral feedback is basically required for exact adaptation. And many circuits, as I said, in biology have exact adaptation. And somewhere inside them, they'll have integral feedback in one way or another. Sometimes it's a kind of hidden integral feedback, but it appears to be found in tissues and inside cells on the level of tissues. We'll talk about examples like that. In glucose, in diabetes control, etc. So this is an important principle. Yeah.
More a biological question, maybe. Say the evolutionary goal is to be where you have more food. Say, so why is exact adaptation more important than
Steady state.
than like the absolute
Yeah.
frequency?
Okay. So why is exact adaptation so important? And so that's the last point I want to make. I want to say, even biologically, if you knock out both R and B, the receptors are in the middle level of methylation. They'll have steady state that looks fine. They don't have adaptation, and if you put a pipette, they can't find the food. So to actually do the search, exact adaptation is more important than steady state tumbling frequency because it allows you to work on a wide dynamic range without being blinded by the food that you have. It's like if you think of a process where you have it's like our visual system, I would say, that if we weren't able to go into a dark room and adapt back, adjust our detectors to low and high level, we'll basically be blind in the dark. So we need to. So those receptors have a limited range where they can detect things, and methylation keeps moving that range to match where the signal is. It's like gain control in a camera. So that's why it's so important when you want in a large dynamic range to adjust your sensor to the proper range. And when you look at individuality in E. coli, this is a very early work, 1976, by Spudic and Koshland. They looked at individual cells, and as I told you, some cells are nervous, and others, genetically identical cells, etc., are relaxed like that. They tumble. Let's say these tumble. Their runs are 2 seconds, and these runs are 0.3 seconds or something. So these ones tumble a lot. These ones tumble a little. So it's genetically identical, same. And if you measure here the steady state tumbling frequency, it varies. And if you measure here the adaptation time, it also varies. This time to adapt, but there's a correlation between them. So these are different individual bacteria. And the model I described here can explain that because these cells are genetically identical, but they have different levels of, let's say, the protein KR, for example. One has 100, another has 150, another one has 80. And this protein, KR, in the model, affects both the steady state tumbling frequency and the response time. It's basically governed by this KR, by how fast you can add methyl groups. And so there's a correlation inside this individuality that's explained by this model. And it allows something, I think, that's what I want to say something about why do you want a population that has nervous and relaxed at the same time? Because it's something quite unknowable for E. coli in advance is whether it'll end up in a liquid that has no obstacles or in a dense situation where there are a lot of obstacles. In dense gel, these ones do better. Because just was like asked before, if you get stuck every half a second, it's good to tumble. But if you don't get stuck every half a second, you get stuck every... If you just have your Brownian motion, it's better to be longer. So this is better. It does it in the liquid here. They do better. So here is this is called bet hedging, which means if you don't know what's going to happen in the future, it's unknowable. It's good to not put all your eggs in one basket and have a diversity within some limits. But all of these have exact adaptation because that works no matter what the protein levels are. And therefore, all of these can work over this 5 order magnitude range. They can reset their sensors if they're at high attractant, low attractant, they can go up vast ranges. And that's how we can rationalize having both differences in bet hedging, being prepared for different situations, and an essential feature, really essential for the key task of navigation, which is this adaptation. No wonder it's in all of our senses. If we want to smell where the cake is, and they don't adapt, then I can't do it. In one circuit. Okay, so here we're taking this idea of robustness. I know this class is challenging, and
so they can also distinguish between a good path and a bad path. Right?
Yes. Yeah.
It's also related to speed, right? Because if it moves faster, then it encounters more signals. So also that.
Okay, the question is speed. If you move faster, everything changes faster like that. So you have to work, even their speed is not very well known because they don't know if they have 2 motors, 3 motors, 5 motors, 6 motors, and speed raises. So the circuit really has to be robust to a lot of unknowables, and it does it brilliantly. So I'm conscious of time, so let's all take a nice deep sigh of relief. And